Hello, my name is Markus Reindl. I'm an associate professor for neuroscience at the Medical University of Innsbruck, and I'm working in the clinical department of neurology. In today's lecture, I will introduce you to the topic of neuroinflammation and demyelination. During the course of this lecture, we will first have a look at the timing of neuroinflammation, then have a look at cellular components of neuroinflammation, the blood-brain barrier, neuroinflammation in neuropsychiatric diseases, and finally, inflammatory demyelination and neurological autoimmune diseases. If you remember what you have learned in immunology on the timing of an inflammatory response, you will remember that you will always need an exogenous, like a pathogen, or an endogenous factor to start an inflammatory response. This can then activate innate immune cells by pattern recognition receptors, for example the toll-like receptor, which you might have heard of, which then gives rise to the activation of innate host defense modules, which within hours lead to the induction of an innate immune response, and then within days to the induction of adaptive immune responses. Here you have two central topics in the immune response: innate immunity and adaptive immunity. And in neuroinflammation, we are mainly talking about innate immunity. Why? It's fast, so it occurs within hours. It's a primary response, which is unspecific and occurs at outer surfaces, and in the case of the nervous system, this is the meninges and the cerebrospinal fluid. It involves tissue barriers, the most famous one being the blood-brain barrier, but perhaps also the blood-nerve barrier, and the cells involved are microglial cells, astrocytes, and macrophages. In contrast, adaptive immunity is lower. It occurs within days. It's a secondary response, which is highly specific, involves T and B lymphocytes, so cells of the peripheral immune system, regulates the innate immune system, and generates immunological memory, which brings us to the cellular components of neuroinflammation. I already mentioned microglial cells and astrocytes before. These two are the most important cells of the immune system or in the brain or in the nervous system. Microglial cells are the tissue-resident phagocytes or macrophages of the central nervous systems, and their specific contribution to an inflammatory reaction is phagocytosis, the degradation of pathogens, antigen presentation by major histocompatibility complex class 1 and class 2 antigens, the production of chemokines and cytokines, and the control of the blood-brain barrier. Astrocytes, what you have heard before in the previous lectures, are involved in neural transmission, but they also play an important role in the immune system of the brain or the nervous system. Because they control the blood-brain barrier, they are important for leukocyte recruitment, for gliosis after an injury, for example, for the production of cytokines and neurotrophins, and also for antigen presentation via MHC class and 2 molecules. Below you find 3 other cell types which you can find in the nervous system which also play an important role in the inflammatory response. Oligodendrocytes, as you have heard, produce myelin in the central nervous system, but they are also important for the inflammatory response because they are a target in inflammatory demyelination, and they can present antigens via MHC class 1 molecules. Endothelial cells, which you find throughout the body, have a specific function in the brain because they build the blood-brain barrier together with astrocytes. They are also capable of antigen presentation via MHC class 1 and class 2 molecules. They can also remove debris, and they can also recruit immune cells. And finally, even neurons can interact with the immune system because degenerating neurons express some MHC class I molecules. And finally, there are also a couple of cells from the peripheral immune system, macrophages, T lymphocytes, B lymphocytes, natural killer cells, and neutrophil granulocytes, which all play an important role in inflammatory responses in the nervous system. Macrophages are important for the control of the blood-brain barrier for leukocytes, antibodies, and complement interaction and cellular and toxicity. T lymphocytes are, of course, important for antigen recognition, for cytotoxicity, and for leukocyte recruitment. B lymphocytes mediate local antibody synthesis in the nervous system. Natural killer cells are important for MHC expression monitoring, for cytotoxicity, and for the detection of viral infections. And finally, neutrophil granulocytes are also important for cytotoxicity, inflammation, and early removal of debris. And here you have already seen that there are two major groups of phagocytic cells in the nervous system, microglial cells and macrophages. So what is the difference between those two populations? It's mainly the origin. Microglial cells have a very early origin because they stem from blood islands in the yolk sac, which then give rise to yolk sac stem cells, then to erythrocyte progenitor cells, which can differentiate to embryonic microglial cells and mature microglial cells. Microglia, as mentioned before, are tissue-resident macrophages of the brain, and as you can see here, they originate from the yolk sac quite early, so they immigrate into the nervous system very early in development. In contrast, macrophages have their origin in the fetal liver and the bone marrow, giving rise to hematopoietic stem cells, then to myeloid precursor cells, to monocytes, which then can differentiate to macrophages and dendritic cells. So this is a kind of second wave of myeloid cells, which then can enter the brain and also play important function there. But the primary immune cell of the brain is the microglial cells. How do microglial cells look like? And this is a very gross schematic overview. Here you have the typical shape of resting or ramified microglial cells. Upon activation, for an example, by an infection, they are then differentiating into reactive or activated microglial cells with this thickened processes here, which then can further differentiate into phagocytic microglial cells, which are at that stage completely indistinguishable from macrophages. And there's also the so-called hyper-ramified microglial cells, which is an intermediate stage between those two, which can also be detected in the brain. In general, if you look at the gross morphology, in a healthy brain, you mainly find this type. In an infected brain, you would find those two, and in an aging brains, this one and this one. What is the function and the activation of microglial cells? Either you have an exogenous factor, like an infection, be it virus, be it bacteria, a tissue injury, which could be a trauma, or an autoimmune reaction. Or on the other hand, you might also have endogenous factors like aging, epigenetics, or genetic factors, all of them leading to the activation of signaling pathways. And as you can see here, there are a couple of them active in microglial cells. Pattern recognition receptors I mentioned before. Toll-like receptors are the most well-known of those. Cytokine receptors, chemokine receptors, neurotransmitter receptors, TREM2 receptor, phosphatidylserine receptor, scavenger receptor, FC receptor, cyclic 3 receptor, and many others more. So there are a couple of signaling pathways which lead to the activation of microglial cells, which then in turn produce cytokines, chemokines, neurotrophins, NO and oxygen radicals, all of them mediating the key biological functions of microglial cells, which are inflammation, cell motility, and phagocytosis. And in a healthy situation, this leads to tissue surveillance, microenvironment scanning, neuronal plasticity, and circuit shaping. All of them things that we wish that they happen. But in a bad situation or a disease situation, this also can lead to autoimmunity and to the acceleration of neurodegeneration. And here you see the classical two-sided phase of inflammation, health, and disease. After having talked about the cellular components of neuroinflammation, I would like to go to the blood-brain barrier in the next step, which is the most important barrier between the peripheral system and the brain or the nervous system. And there's a schematical view how it looks like in vertebrates. You have a capillary with immune cells in there, and then a layer of endothelial cells which are connected by tight junctions in here. And what is specific or different of the blood-brain barrier is the basement membrane, shown here in yellow, which is formed by pericytes, cells specialized for this function in the nervous system, and the glia limitans, which is formed by footpads of astrocytes, both of them giving the blood-brain barrier a very tight appearance. Of course, there are also neurons connected to it, microglial cells and macrophages present in the perivascular space. So to summarize, the blood-brain barrier consists of an endothelial cell lining with tight junctions, and an endothelial and a parenchymal basement membrane, the glia limitans formed by astrocyte processes linked by gap junctions, and finally microglial cells and other antigen-presenting cells. Blood-brain barrier is very tight, so water-soluble agents, of course, can pass it very easily via tight junctions, shown schematically here, where you have a water-soluble agent which can pass the blood-brain barrier through the paracellular aqueous pathway. Lipid-soluble agents also can pass the blood-brain barrier quite easily through the transcellular lipophilic pathway. And this is also used for drug development, where drugs are either water or lipid-soluble, so that can pass the blood-brain barrier. With higher molecular weight molecules, it's more difficult because small molecular weight molecules like glucose, amino acids, and nucleosides use transport proteins like the glucose transporter to be transported through the blood-brain barrier. Larger molecules like insulin, transferrin use receptor-mediated transcytosis, shown schematically here, where you have the transferrin receptor binding to transferrin, which is picked up by the endothelial cells and then transported to the other side. Albumin and other plasma proteins are transported by absorptive transcytosis, shown here in a similar way. So in general, blood-brain barrier is very tight, and you need specific pathways to pass it, which is a major problem for drug development. How can lymphocytes and other immune cells pass the blood-brain barrier? Therefore, they need adhesion molecules which mediate tethering, which is the adhesion to the endothel, rolling, and transmigration of monocytes and lymphocytes through an activated endothelium. And there are three major families or classes of adhesion molecules: the selectins, which bind carbohydrates, initiate leukocyte endothelial interaction, for an example p-selectin, e-selectin; the integrins, which bind to cell adhesion molecules and extracellular matrix, mediate strong adhesion. One example is shown here, the VLA4 or alpha-4 beta-1 integrin. And finally, the Ig superfamily, which has various roles in cell adhesion and which is a ligand for integrins. Example shown here is VCAM. So a lymphocyte expressing VLA4 will meet an endothelial cells expressing VCAM. They will attach, then they will roll, and finally they will enter the brain parenchyma via diapedesis, leading to leukocyte infiltration and brain inflammation. And here I have a summary or some videos where you can have a closer look at this. All of them available at the homepage as shown here, which show nicely the adhesion of immune cells to the blood-brain barrier and the transmigration. Which cells can enter the nervous system after activation of the blood-brain barrier? In the normal brain, you only find very little in the invasion of peripheral immune cell. You find some T cells which patrol the central nervous system for the typical immunosurveillance of the brain to detect virus infection. The same holds true for NK cell, natural killer cell, and macrophages. If there's an indirect trauma, you find more T cells and NK cells. In the case of neuronal cell death, this frequency is increased even more. In the case of a direct injury, be it an open traumatic brain injury or spinal cord injury, macrophages, granulocytes, and T cell can enter the nervous system. And finally, in the case of an infection or autoimmune inflammation, you see a lot of cells entering the nervous systems, T cells, macrophages, granulocytes, and even B cells. So then we come to the next chapter, which is neuroinflammation in neuropsychiatric diseases. I think all of you have heard about that before, and it's currently investigated in many neuropsychiatric diseases, whether neuroinflammation plays a causal role there or whether it plays any role there. In general, we distinguish between primary neuroinflammation and secondary neuroinflammation. Primary neuroinflammation is seen in neuroinflammatory disorders, for an example, multiple sclerosis, Guillain-Barré syndrome, autoimmune encephalitis, and I will talk about this immediately. On the other hand, secondary neuroinflammation is seen in almost all neurodegenerative diseases, such as Alzheimer's disease, Parkinson's disease, multiple system atrophy, amyotrophic lateral sclerosis, but also in psychiatric diseases such as depression or schizophrenia. The main difference is that in these diseases we think that the immune response directly leads to nervous system tissue damage, whereas here it's a secondary phenomenon or it's a consequence of primary neurodegeneration and primary nervous system damage. The neuroinflammatory diseases can be, if we talk about neuroinflammatory disorders, we talk about inflammatory demyelinating diseases, autoimmune encephalitis with antibodies against neuronal surface antigens, autoimmune encephalitis with antibodies against intracellular neuronal antigens, and neuropsychiatric SLE. Inflammatory demyelinating diseases, the most famous and frequent disease is multiple sclerosis, but there are also rare disorders like neuromyelitis optica or acute disseminated encephalomyelitis, and Guillain-Barré syndrome and chronic demyelinating polyneuropathy of the peripheral nervous system. Autoimmune encephalitis with antibodies against neuronal surface antigens compromises a variety of rare disorders which are all characterized by specific antibodies against neural receptors, the most famous being NMDA receptor encephalitis, but there are of course many others, as you can see here. This list is ever growing, and at the moment every year 1 or 2 new disorders are discovered. In the case of autoimmune encephalitis with antibodies against intracellular neuronal antigen, this mainly indicates a CD8-mediated T-cell response against those antigens, and they often occur in a paraneoplastic context with a tumor expressing the antigen also present in the nervous system and then leading to an autoimmune response. And finally, lupus can also manifest within the central nervous system or nervous system as neuropsychiatric SLE. Inflammatory and demyelination and neurological autoimmune diseases will then be the last and major topic of my lecture. And if you look at acquired inflammatory demyelinating diseases, we distinguish those that affect the central nervous system and those that affect the peripheral nervous system. In the central nervous system, the most frequent one is multiple sclerosis, but there are also atypical rare forms like NMO, spectrum disorders, ADEM, acute multiple sclerosis, Pallo's sclerosis, and others. In the peripheral nervous system, we have an acute form, Guillain-Barré syndrome, with its specific form, Miller-Fisher syndrome, and the chronic inflammatory polyneuropathy form. Before I go into detail, I just have to repeat in briefly myelin structure and function. You have here schematically a neuron in the central nervous system with axons which are surrounded by myelin sheaths produced by oligodendrocytes, and myelin nodes, internodes, are separated by the so-called nodes of Ranvier, which are crucial for signal transduction. Neuronal signals in mammals or in vertebrates have to be conducted over long distances. In man, it's over 1 meter. Continuous signal transmission is very slow, and the mere increase of axon diameter does not allow a significant increase in conduction velocity. Therefore, nature has invented myelin because myelinated isolated axons, the so-called internodes shown here, are impermeable with low capacity. And we have a saltatory conduction or so-called saltatory conduction by the nodes of Ranvier, shown here, where there is a high density of sodium and potassium channels. Myelin sheath thereby increase conduction velocity, and demyelination leads to an increase of membrane capacity. Sodium currents are too low, and there is no more or diminished propagation of action potential. And this is the reason why demyelination or dysmyelination leads to such severe neurological deficits. Here are some examples of demyelinating disorders. In the case of inflammatory demyelination, we currently propose that this is caused by autoimmunity, which either includes epitope mimicry or an abnormal immune regulation. Example of human disorders would be multiple sclerosis, the Guillain-Barré syndrome, and chronic inflammatory demyelinating polyradiculoneuropathy, CDP. There are also rodent models for this, the so-called experimental autoimmune encephalomyelitis EAE model, or the experimental autoimmune neuritis EAIN model. In the case of an injury to oligodendrocytes or Schwann cells by trauma, viral infection, apoptosis, or necrosis, this can be seen in the brain, the spinal cord, and upon nerve injury, but also upon ischemia, and for infection, for example, in the case of progressive multifocal leukoencephalopathy, BML. Rodent models would be a step wound lesion, a contusion injury, and nerve crush, a toxic injury by cuprison or ethidium bromide, middle cerebral arterial occlusion, commonly used model for stroke, or Tylers and mouse hepatitis virus infections. And finally, there are also primary demyelinating disorders caused by genetic defects of the oligodendrocytes and Schwann cells, including myelin gene duplications, point mutations, and lipid storage disorders. Examples are Pelizaeus-Merzbacher disease, Charcot-Marie-Tooth disease, adrenoleukodystrophy, and metachromatic leukodystrophy, all of them being rare disorders, fortunately enough, mostly or almost all of them affecting young children. There are also corresponding mouse mutants, either spontaneous or generated by gene targeting, which are used to model these diseases. Multiple sclerosis, as mentioned before, is the most common of all these demyelinating disorders, and it's a major cause of disability in young adults in Europe and North America. The prevalence is about 100 per 100,000, so 1 in 1,000, which is quite large. Worldwide are currently 2.5 million individuals infected. It's a disease that mainly affects females. Females to male ratio is 3:1, and the prevalence of females is increasing over the past decades. Typical age of onset would be between 20 and 30 years, so young adults are mainly affected by the disease. It's a disease that mainly affects Caucasians, so the highest prevalence is seen in Caucasian. It's common in Northern Europeans, and of course also in the USA or Canada, intermediate in Asians and the Orient, and rare in Africans, which points to the influence of environmental and genetic factors. MS plaques or MS lesions may form in the central nervous system white matter, but also in the gray matter at any location. Therefore, the clinical presentations of the disease are diverse and affect all neurological symptoms. The continuing lesion formation in this disease often leads to physical disability and also not infrequently to cognitive decline. This graph shows a typical disease course in multiple sclerosis patients. So usually patients start with a so-called clinically isolated syndrome, which is the first manifestation of multiple sclerosis. Then after time enter a so-called relapsing remitting disease course, which is characterized by disease relapses and phases of remission. And finally, after time, about a half of the patients enter a so-called secondary chronic progressive disease course with accelerating disability. Over the time, inflammation is very high at the beginning of the disease and is declining. In contrast, axonal loss is accelerated over the course of the disease, rare at the beginning and very frequent at the chronic stages of the disease, which is also reflected by the loss of brain volume. At the beginning of the disease, you have dominance of inflammation, demyelination, axonal transaction and remyelination. After time, continuing inflammation and persistent demyelination, and in the end stage you have a sequestered inflammation, chronic axonal degeneration and gliosis. And here the disease turns from a peripheral immune disease to a central nervous system immune disease, to a more chronic disease which has many similarities with neurodegenerative diseases then. And all of this is a function of age, with inflammation being most prominent in young age and degeneration dominating in older individuals. MS pathology is quite heterogeneous, so in the early inflammatory stage you have an important role of cells of the peripheral immune system like B lymphocytes, CD4 and CD8 positive T cells, and at least 3 different patterns of demyelination have been described in focal white matter lesions. Some of them are dominated by cytotoxic T cells, macrophages, some of them by antibody-mediated demyelination and macrophages, and some of them by oxygen and nitric oxide radicals produced by macrophages, which lead to a hypoxia of oligodendrocytes. In any way, after time, there's a common end road of the disease, which is chronic persistent inflammation and persistent microglial activation with diffuse demyelination, axonal injury induced by microglia toxins like oxygen and nitric oxygen radicals, cytokines, and mitochondrial damage, which leads to diffuse injury in white matter and subglial cortex, observed in chronic progressive disease. There's a debate going on now for more than 100 years whether multiple sclerosis is an autoimmune disease. Why? So generally, we think that MS is currently considered to be a T-cell mediated autoimmune disease. This view is based on the one hand on the cellular composition of brain and cerebrospinal fluid infiltrating cells, so from the neuropathology of the disease, and on the other hand from data from the animal model experimental allergic autoimmune encephalitis. In this model, the injection of myelin components into susceptible animals lead to a T-cell mediated autoimmune disease which shares many similarities with MS and which can be transferred to other animals by encephalogenic T-cells. There are also several genetic studies which support the role of T-cells in MS, genetic association studies which show that the strongest genetic link for multiple sclerosis is a specific HLA class II allele, the so-called TR2 or TRP1115 allele. And there's also association with several genes of the inflammatory response, but not of the nervous system of neurodegenerative processes. And finally, and most importantly, all currently used treatment and experimental treatments also support the understanding of an MS as an autoimmune disease. The ERE model, as I mentioned before, can be induced in susceptible experimental animals by active immunization with CNS homogenates or myelin components. Examples are shown here: myelin oligodendrocyte glycoprotein, MBP, and others. Importantly, this only works if you use a strong adjuvant like complete Freund's adjuvants. By the passive transfer by in vitro activated autoimmune T-cell lines, and finally in the past year also spontaneous ERE models with transgenic mice in which a large proportion of T-cells or B-cells are myelin specific have been generated. They are susceptible and resistant rodent strain, but in each animal the model or the disease strongly develops on the nature of the autoantigens. Some autoantigens are highly encephalogenic in one strain but not in the next. Examples are shown here. For example, Lewis rats and plaque 6 mice have either self-limited monophasic or chronic disease, and there are also relapsing remitting diseases which are shown in SGLJ mice, Biopsy mice, or BN rats. So there's a large heterogeneity which also reflects the human situation where no MS patient is like the other. MS has a familial recurrence rate of about 20%, so about 20% of all affected patients are seen in families. The relative risk is about 30% in monozygotic twins, 3% in first-degree relatives. In siblings it would be 5%, in parents 2%, in children 2%, 1% in second-degree relatives, and 0.1% in the general population. So obviously there is a genetic component which does not cause the disease, but which modifies disease risk. The association between MS and specific MHC HLA alleles were identified already in the 1970s, and it has been in the past year shown that the most important allele is the TRP1 1501 allele, which increases the risk to develop multiple sclerosis dramatically. In the past 10 years, also several genome-wide association studies have been undertaken in currently more than 30,000 subjects with MS and 60,000 controls. We have about 110 established MS risk variants at 103 discrete gene loci, all of them outside the MHC complex, and all of them are variants of genes associated with immunological processes which are located in regulatory rather than coding regions, and which are frequently associated with other autoimmune diseases. So far, no association with genes relevant for glial or neuronal function have been found. Another clue for multiple sclerosis comes from the distribution of the disease, which I have already mentioned before. You can see here nicely the World Atlas of Multiple Sclerosis with the highest prevalence found in Europe, Northern America, and Australia, so in regions where you have a predominance of Caucasian populations, also high frequency in the former Soviet Union and the lower frequency at the equatorial region. Established environmental factors so far are Epstein-Barr virus infection, vitamin D and sunlight, which fits nicely also to the distribution shown here, smoking and diet. And currently we think that MS is a complex disease mediated by environmental factors, pathogens, smoking, sunlight, vitamin D, diet and chemicals, epigenetics and genetics, all of them interacting to increase the susceptibility of for multiple sclerosis and modify the disease course. And this is a very, very gross and simplified scheme how we think that the immune pathogenesis of MS happens. You have a peripheral infection which leads to the activation of autoreactive T cells. These autoreactive T cells can then pass through the activated blood-brain barrier and within the central nervous system lead to inflammation and demyelination by activation of microglial cells, of macrophages, also of B cells, then to demyelination and axonal damage. And finally, as I mentioned before, also all currently used treatments for multiple sclerosis are anti-inflammatory, which you can see here. This is just the current recommendation for these treatments. We have a treatment of relapsive with corticosteroids and plasma exchange, all of them only affecting the immune system, and then a so-called interval treatment with treatment escalation with increasing disease activity, starting from beta interferons up to monoclonal antibodies against adhesion molecules. For an example, Diasabri is at the moment by far most effective treatment for multiple sclerosis. As mentioned before, all of these treatments are anti-inflammatory. None of them affect the nervous system. There is also symptomatic treatment like neurorehabilitation and neuroprotection, neuroregeneration, which plays an important role, but which does not modify the disease course. Last but not least, I would like to introduce you to the diagnosis of multiple sclerosis and other inflammatory demyelinating diseases. In any case, you have a first demyelinating event. You can never diagnose the disease only by one event. You always need a dissemination in space, which means the neurologist has to detect two MS lesions in two different localizations within the central nervous system. Also, dissemination in time is required, which means two relapses or a delayed MRI with active MRI lesions. Inflammation and of course the exclusion of all possible differential diagnoses, and for multiple sclerosis these are more than 100. This is done mainly by the neurologist, by the clinical history of the patient, by careful neurological examination, by magnetic resonance imaging, cerebral and spinal, which is able to detect specific MS lesions, by cerebrospinal fluid investigation, at least in Europe. And finally, also autoantibodies are used for the differential diagnosis of the disease, as you can see immediately here, because after this investigation, the neurologist would either diagnose possible MS. In this case, he would have to wait for a second relapse or a delayed MRI, definitive multiple sclerosis, a so-called atypical demyelinating disease like ADEM or neuromyelitis optica spectrum disorders, or finally another neurological disease. Why are these aquaporin 4 antibodies so important? They are a diagnostic hallmark of the disease NMO, neuromyelitis optica. This is a severe bilateral optic neuritis and myelitis, which occurs monophasic or in relapses in short time intervals. The disease was initially described by the French neurologist Eugène de Vic in 1894. It's a rare disorder in compared to MS, so it affects about 1 in 100,000. If you remember, MS affects 100 in 100,000. It's a disease that mainly affects females, so more than 80% of affected patients are females, and it has, if not treated correctly, a poor prognosis. According to the old diagnostic criteria, this involves mainly clinical criteria, optic neuritis, myelitis, and supportive criteria, brain MRI not meeting criteria for multiple sclerosis at disease onset. And in the spinal cord, you see this very prominent contiguous T2-weighted signal abnormality, which extends over 3 lesions, so-called longitudinally extending transverse myelitis. And finally, the so-called aquaporin 4 antibody seropositive status. So if patients have these autoantibodies, the disease can be diagnosed, and in the new diagnostic criteria published last year, this now is one of the major diagnostic criteria. Having this said, I will conclude the chapter on demyelinating diseases and finally come to autoimmune encephalitis with antibodies against cell surface and synaptic proteins. This is a disease entity which became more prominent the past few years due to the fact that every year 1 or 2 new autoantibodies are described in the nervous system. And you see here a list about the most prominent or the most frequently seen diseases and autoantibodies, the most prominent of them being the NMDA receptor encephalitis, which is a disease that is monophasic, affects young adults and children with a female preponderance, and is characterized by psychiatric, memory and motor deficits, by seizures and autonomic instability. Often it's a paraneoplastic disorder, so it can be observed after teratoma, ovarian teratoma, and it responds to immunotherapies. And from the point of view of neurobiology, this is a very interesting disease because, as you have heard in previous lecture, the NMDA receptor is crucial for memory formation, for synaptic plasticity, and in this disease, autoantibodies target this receptor. The receptor is targeted by them, and then it's internalized, and this leads to a deactivation of the receptor. It's just not present there on the surface again, and then leading to all these severe neuropsychiatric deficits. And it also leads to the impairment of memory and behavior in vivo. This is a disease that in earlier times was mainly thought to be a psychiatric disease. Now we know, when we can diagnose it with the antibody and treat it with immunotherapies, patients will recover, which is not the case if they are not treated in this way. Other examples, receptors, AMPA receptor, GABA-B receptor, components of the voltage-gated potassium channel, and many other. The most or the important method is just here that against almost every receptor of the central nervous system, and meanwhile autoantibodies have been found. And this is summarized in the last slide here, that different autoantibodies which can originate or be associated with psychiatric disorders like NMDA receptor antibodies, AMPA receptor antibodies, with epilepsy like GABA-B receptor antibodies or NMDA receptor antibodies, limbic encephalitis, voltage-gated potassium channel subcomplexes, LG1 and CASPA, paraneoplastic antibodies, even after a viral infection, after herpes encephalitis, NMDA receptors can be built. And finally, atypically demyelinating diseases can also lead to an alteration of synaptic function, where you have severe neuropsychiatric diseases which originate from the immune system. And with having this said, I will conclude my lecture.